So this is our PowerPoint about enzymes. I've been talking about enzymes for quite a while with you. It's sort of come up in different ways. We've talked about enzymes when we were talking about reactions that happen in living systems. So all reactions in living systems, or pretty much all reactions in living systems, are catalyzed by enzymes. And we talked about enzymes again when we talked about proteins and the fact that enzymes are composed of proteins, sometimes with a little bit of extra stuff thrown in, but primarily protein in, the majority of the structure is protein in nature. So we're going to have a look at some of the characteristics of enzymes and some of the interesting bits and pieces about enzymes. Be sure you have your textbook handy because there's a few questions that are going to come at the end of the sheet that aren't covered by this PowerPoint that you're going to have to look up yourself. So what are enzymes? As I mentioned already, most enzymes are proteins, and in order to be functional, you know that they must have reached tertiary or quaternary level of organization. The enzymes in our bodies, which is one of the topics not covered on this little PowerPoint, the enzymes in our bodies are generally what we call allosteric enzymes. And those enzymes are enzymes that have achieved the quaternary level of organization. They're composed of two polypeptides, one which has an active site and one which has a control site for either activating or inhibiting the enzyme. So turning it on, turning it off when we need it. The enzymes act as catalysts, which means that they are going to help carry out our reactions more effectively. One of our limiting factors in biological systems, one thing that we have to keep in account or take into account, which we can't, or that we don't have to when we're working in the lab, is that we obviously can't raise our temperature beyond that can be tolerated by the system. So unless you're a hot spring bacteria or something like that, we've got real limits about the temperature range that we can operate those chemical reactions, where in the lab we often will add heat to catalyze the reaction to carry on, to move in the forward direction. These enzymes, the catalysts, are not permanently changed in the process, so there is a conformational change, meaning the shape does change, but after the reaction occurs, the enzyme goes back to its original shape, the shape that it must be in order to accept the next substrate into its active site. So I know this little graphic looks like a great big blob, but this is not this is not uncharacteristic. In fact, it's very characteristic of the majority of the enzymes you have in your body. They look like blobs. Again, that shouldn't really come. So as I was saying, sorry about that. As I was saying, this would be characteristic, and this will have achieved at least the tertiary level of organization, most likely the quaternary level of organization. You can see this white area in here would be the active site. So what looks like a blob is in fact shaped exactly right to be able to accept whatever the substrate is into the white area in the diagram. Enzymes are specific. So in our bodies we have as many enzymes as we have reactions. They won't catalyze, or they generally won't catalyze a reaction that they're not designed to catalyze. As I mentioned earlier, once they've carried out their task, once they've conformed to the new shape to allow whatever the reaction is to occur, they will resume their original shape and then they're ready to go again. A lot of the enzymes end in the word ase, and we might have mentioned that in class with things like lactase, the lactase enzyme, and a few other things that we talked about in class. So if you see ASE in the end, most likely you are dealing with some kind of an enzyme. And here's a couple of examples: sucrase, lactase, and maltase. And on your sheet, I'd like you to take a stab at guessing what reaction those particular enzymes might catalyze. I think it's pretty obvious, but give it a try, and we'll talk about it in class. So our little graphic here is just showing a green colored enzyme with an active site and some kind of substrate. So that's the material that's going to enter the enzyme to start to be the reactant to whatever's reaction is going to happen. And again, the shape of the active site is just the right shape for that substrate to fit in there. So enzymes, because they are going to sort of stretch the bonds or press pieces of particular atoms together, they will either weaken bonds or they will allow bonds to form more easily. So the activation energy for the reaction to occur is lowered, which again is a good thing because we don't want to have to add a whole lot of heat to get things moving. Again, we're looking at a little graphic here, and it's showing that the enzyme is able to lower the activation energy for this reaction to occur, for Y to go through whatever the reaction is to become X the product. And instead of having to add all the energy to go up to D, a catalyst would sort of make it sort of a B level, so it would be a lot easier for the reaction to take place, would take a lot less energy. And here's a nice little graphic that shows that. This is the one that you might want to copy onto your sheet where it asks you to do so. And we've got reactants and products. And we're going to see two lines are going to be drawn on here without an enzyme and with an enzyme. And if you look at the labels for the graph, we can see the progress of reaction is charted along the bottom and free energy is charted along the side. So the reaction without the enzyme is our blue line that's just been drawn on there. And the pink line is our reaction with an enzyme. And again, you can see that there's quite a difference in the amount of energy that is required to make that reaction occur. Here's an example of an enzyme catalyzed reaction. This is hydrogen peroxide. That's the substrate, the material that's going to undergo the change. And in the first test tube on the rack, we've got the enzyme without the substrate, so nothing is happening. But in the second, we've got an enzyme, plus we've got our hydrogen peroxide. And the enzyme that we're using here is a liver homogenate. So they've ground up some liver, and there's a catalase enzyme in there. So it's going to, catalase enzymes break down hydrogen peroxide, which is toxic to the body, into water and oxygen. And what we're seeing in the or rather they catalyze that reaction, I should say. What we're seeing in this test tube is the product oxygen. So we're seeing the bubbles forming, the gas being given off there is oxygen gas. So that's just an example of an enzyme catalyzed reaction, and that happens in our bodies where our liver is able to, we don't homogenize ours, but our liver in our body is pardon me, is able to break down hydrogen peroxide. All right, so I've been using that word substrate, and here's where we get a look at the definition of that. So whatever the reactant for the reaction is, if we're talking about the enzyme, we refer to it as a substrate. So the substrate is the reactant that the enzyme is going to enable to go through whatever the reaction is that needs to happen. So here's a nice graphic of a green enzyme, and we've got our yellow substrate, and that substrate will fit right into that sort of square shape where it looks like it's meant to go, and then all kinds of things are going to happen to help the reaction occur. So the area where the substrate binds to the enzyme is called the active site of the enzyme. And it's very specific, again, in shape. And that shape, remember, is going to be due to all kinds of interactions, a primary, secondary, tertiary, and likely quaternary level of organization of the protein. And sometimes there's even some other things that are kind of chucked in there with the enzyme to help them work. So what seems like a glob is actually very specific in nature. And the active site, again, particularly designed to fit the substrate. So we used to think that enzymes and substrates fit together like a lock and key, kind of like the substrate into the enzyme, and then it would somehow turn or start, I don't know what we thought it would do, but would just sort of lock into place, the substrate would. And the thought now about how this enzyme, sorry, pardon me, I hope I don't repeat myself. So now what we feel happens is that the basically that there are bonds that are formed between the active site of the enzyme and the substrate, whatever it is. So there could be ionic, there could be more likely covalent and hydrogen bonding that occurs there. So that the enzyme is able to put strain on a bond that's going to be broken, or is able to flex in such a way that two parts that need to be joined together are sort of closer together, for lack of a better way to put it, and a bond is easy to form between them. So when the substrate fits into the active site, the attachment or the bonding, physical or chemical bonding that happens there, most likely, again, more physical bonding, because that's easy to make and break. When that happens, or that interaction is going to be what helps move the substrate in the direction for the reaction to take place. So the shape that enzyme substrate complex, the shape is sort of induced by the substrate being fit into the active site. So again, the substrate traveling into the active site of the enzyme causes a change, a configural or a conformational change in the active site of the enzyme, which sort of makes whatever the reaction that's coming is going to happen a little bit easier. And saying here that it's possibly hydrogen bonds or ionic bonding that are involved there. So the shape change is induced by the substrate. The shape change happens when the substrate fits into the active site, and is going to allow whatever the reaction is to occur after that. Well, we know that enzymes are proteins for the most part, so I think you could probably guess what the three factors are going to be that might affect enzyme activity. They could be environmental conditions, cofactors and coenzymes, and enzyme inhibitors. Really, I guess you wouldn't be able to guess, maybe 2 and 3, but environmental conditions, certainly you should be able to figure out what conditions we would need to maintain to keep the proteins happy. For instance, we'd need to make sure that the pH was okay for whatever the enzyme might be, and that the temperature is within the range that they are functional, and also the salt concentration or ion concentration in the environment, same as it would be for proteins. So we'll have a look at the other bits and pieces here that you may not be familiar with. So as I said, temperature, pH, and salt concentration have to be within range, or we risk having the protein denatured. Cofactors, I've sort of hinted at these, cofactors and coenzymes. Sometimes the enzymes require some other things to be able to work a little bit better. So sometimes the enzyme will require either some inorganic or organic substances for proper activity. So inorganic substances, perhaps like zinc and iron, would be considered to be cofactors, and vitamin derivatives would be considered to be coenzymes because they are organic in nature. And again, they're often needed to aid the enzyme in maintaining the right shape to be able to do the right job. All right, now I've already mentioned that we can control our enzymes in many cases by either inhibiting or activating the enzymes. So our living systems have that capability. Sometimes inhibitors, though, are things that are not things that we've made to try and control those enzymes, but could be things in the environment that are competing or that change the shape of the enzyme. So there are two types of inhibitors. They can be a competitive inhibitor, which will often look very much like or be very similar to the shape of the normal substrate that should fit into that active site. And I'm sure you can figure out what's happening there. So if it's a competitive inhibitor, what's going to happen is it will occupy the substrate's normal active site so that the substrates can't get in there. All enzyme activity, this is actually a really important point and it's not on this PowerPoint, all enzyme activity is a function of concentration. So how much substrate is there, what is the concentration of the enzyme in the environment, those two things are going to have a huge impact on how many reactions are going to be carried out. So if we don't have much substrate, then the reactions can be catalyzed quite merrily along. If we had a competitive inhibitor or any kind of inhibitor, something that was stopping the enzyme from working, no matter how much substrate we have, we still won't be able to carry out those reactions. So concentration of both of these things is key to how many reactions are going to be occurring. So we've got competitive inhibitors, which would be like this little illustration. Say that the blue block is fitting in instead of the yellow substrate to the active site, and now the substrate can't get in there because the competitive inhibitor is kind of blocked in there. Non-competitive inhibitor don't enter the active site but have some other binding site on the enzyme, and by binding there it's going to change the three-dimensional structure of the protein, and the active site's not going to be the right shape anymore. So we're seeing here, same green enzyme, there's a non-competitive inhibitor, and our active site is no longer the right shape, and the substrate will not fit in there. So we tend to use non-competitive inhibitors a lot to turn off our own enzymes, but there can be outside inhibitors that are going to turn those enzymes off. All right, we've reached the end of this little PowerPoint, but there are a few other things on the bottom of your sheet that I want you to look up in your textbook or online. One is allosteric enzymes, which I mentioned already in this PowerPoint presentation, but you should probably look up to find a little bit more detail about it. The second thing is multi-enzyme complex, which is going to be very important for us in our metabolic functions unit. And again, I think partly the name gives it away, a multi-enzyme complex. So you can look up a few examples, look at some details about that. And the last one that I've asked you to look at is something called feedback inhibition. And that's going to be essential when we talk about our multi-enzyme complexes. So those two things sort of go together. And bring any questions that you have to class.